The human brain is the most complex living structure in the universe. It is inside the skull, which is important because it protects the brain from impacts, suspended in cerebrospinal fluid and isolated from the bloodstream by the blood-brain barrier. The layers that are between the brain and the skull are referred to as the meninges. These are the dura mater, the arachnoid, and the pia mater. The thickest and outermost layer is the dura, which is almost like a thick sheet of leather. All of them protect the brain and keep the cerebrospinal fluid from leaking.
The brain has two main parts, a right and a left cerebral hemisphere. Both of them are mirror images of each other, and they are connected by the corpus callosum. The structure of the brain is associated with specific functions in the body. Each hemisphere has specialized areas which are responsible for motor movements, sensation, thinking, and seeing. The brain has four lobes. The frontal lobe, which is in the front region of the brain, is in charge of motor movements, which is the motor cortex. There are also other parts that are important for the ability to speak, and this area is referred to as Broca's area. And other parts of the frontal lobe are also in charge of cognitive skills. Behind the frontal lobe, it is the parietal lobe. There is an area in this lobe called the somatosensory cortex, which is very important for the ability to interpret sensations from the body. Then we can find the occipital lobe that is in the back part of the brain, and it is important for vision. The temporal lobe, which is beneath the sylvian fissure, is important for hearing, and this area is called the auditory cortex. This lobe is also important for short-term memory.
However, the human brain has not always been as it is today. It is the result of a long process of evolution. After 7 million years, the human brain has increased in size. Nowadays, the brain is 3 times bigger than it was before. Even though there are no ancient brains to weigh on a scale, the inside of ancient skulls can be measured. Unfortunately, a few fossils have preserved natural shapes of the interior of skulls. For the first 2/3 of our history, the size of our ancestors' brain was within the range of those of other monkeys living today. For example, Australopithecus afarensis had skulls with internal volumes of between 400 and 550 milliliters, whereas chimpanzee skulls hold around 400 milliliters and gorillas between 5 and 700 milliliters. During this time, Australopithecine brains started to show changes in structure and shape as compared with apes. For instance, the neocortex had begun to expand, reorganizing its functions away from visual processing towards other regions of the brain. The final third of our evolution saw nearly all the action in brain size. Homo habilis, the first of our genus Homo, saw an increase in brain size, including an expansion of a language-connected part of the frontal lobe called Broca's area. The first fossil skulls of Homo erectus had brains averaging a bit larger than 600 milliliters.
From here, the species brain started to rise, reaching more than 1,000 milliliters by 500,000 years ago. Early Homo sapiens had brains within the range of people today, averaging 1,200 milliliters or more. As our cultural and linguistic complexity and technological abilities took a significant jump at this stage, our brains grew to accommodate the changes. The shape changes are related to planning, communication, problem solving, and other more advanced cognitive functions. With some evolutionary irony, past 10,000 years of human existence actually shrunk our brains. One of the reasons of this shrinking is the limited nutrition in agricultural populations. On the contrary, industrial societies in the past 100 years have seen brain size rebound as childhood nutrition increased and disease declined. Although the past does not predict future evolution, a greater integration with technology and genetic engineering may catapult the human brain into the unknown.